In this video, we're going to be talking about laboratory techniques. Now remember our central dogma: DNA to RNA to proteins. And most of the time you go through that cycle, that principle fairly well, no problems there. But sometimes there are problems, there are mutations. And we want to look for those mutations. We want to look to see if there's a problem with any of these. Why? Because if there's a problem, you can have things like genetic disorders. Another reason we want to look for abnormal RNA, DNA, proteins, etc. is, how about if you have an infection, like a viral infection? We want to look for viral proteins or viral RNA. And lastly, something less sinister, if you want to do research and you want to see what a particular protein does or a strand of RNA does, you want to isolate that. So we'll just say research. Either way you cut it, it's very important to understand how we isolate DNA, RNA, and proteins. And that's the subject of this video. It's all about lab techniques. Lab techniques. And these will be in no particular order. So the first one I'm going to start with is perhaps the most famous, and that is PCR, or Polymerase chain reaction. This is when you have a piece of DNA that you want to study, that you want to make more copies of. Why do they call it polymerase chain reaction? Well, you're going to put a polymerase on here and you're just going to photocopy a ton of these copies. That's the chain reaction part. And then at the end, you're going to have times whatever amount, just tons and tons of the DNA in question. How do we get to that point? Well, DNA is tightly annealed, as a double complex is tightly held together. So we want to first separate the DNA. That is called the denaturing process. That's just done through heat, usually at around 95 degrees Celsius. You heat it up and it'll start to separate. So now it's a little bit more separated. That'll be our sequence in question. Second step is called annealing. Not of the two strands back together, but instead annealing of a DNA primer. So you put a DNA primer on here. You want to cut the heat, otherwise the DNA primer would never stick on. That too would get denatured. So you want to cut the heat. Now you're down to around 55 degrees Celsius. And the last step is going to be your elongation. This is when you have your primer, you put on a DNA polymerase on that primer, and it'll start to make your copies of your DNA. Now we're at 55 currently, and we put that on. If we keep cooling it, then the whole thing will anneal and you won't be able to process it. So we usually heat it back up to around the midway mark, we'll say 72. But again, that can vary. But we heat it to the midway mark. That way it's right in that, I guess, the Goldilocks zone, where it's not too cold where it anneals and not too hot where everything denatures. So around 72. So know the steps, roughly know the temperatures, and know why it's called polymerase chain reaction. You put a polymerase on, just start photocopying. That's the first one. I'll leave that on there. The second one I want to talk about is actually a continuation of the first. You have one sequence you want to make, but you can also do it with multiple sequences. And if you have those multiple sequences and you make a ton of copies, how do you separate them? You separate them by what we call gel electrophoresis. And this isn't only to DNA, this can be done for RNA, proteins, whatever your central dogma is. And you separate them by size and you separate them by charge. How do you do that? you have a little gel plate, and one side is negatively charged and one side is positively charged. And you put all your different segments of your DNA, protein, RNA, etc., and you run that current. And like I said, they separate by charge. So things that are very negatively charged will want to move on this end. You separate them by size. So things that are small, easy to move, again, will move towards this end. Eventually, you'll have nice separated segments, and you can tell and distinguish them apart. That's just gel electrophoresis. That's just used to separate whatever you need to separate. Keep that in mind. We're going to be referencing gel electrophoresis. That's why I wanted to talk to you about it this early on. And when I say reference gel electrophoresis, I mean immediately. So the next thing we're going to talk about is blots. Blots, again, will look for things in your central dogma. So if you want to look for a particular DNA sequence, we have a blot for that. If you want to look for a particular RNA sequence, we have a blot for that. Protein sequence, special blot for that. The mnemonic that is on first aid, and the one that I like also, and I use it quite a bit, is snowdrop. The snow is named after the name, so southern, northern blot, and western blot. And then the drops is named after what they detect. So it's going to be your DNA, RNA, and your protein. So your southern blot detects DNA, northern blot detects RNA, and then western blot detects protein. Why do they call them blots? Well, we'll just take southern for example. The reason they call them blots is to understand why they call them blots, you're going to need to understand how blots are performed in the first place. The first step in blots is you will separate the DNA by enzymes, because that's what we're looking for in the southern blot. So separate DNA by enzymes, then run it through electrophoresis to kind of organize your pieces. Once you separate it, it's still on that electrophoresis gel, and that's just gel is not very stable. So you want to stabilize it by putting a piece of filter paper on top. You kind of blot the gel. And when you blot the gel, the DNA will move onto the filter paper. So we'll just say, second step, you just blot the gel with filter paper. It's a little bit more complex than that, but that's essentially what you do. And the DNA will migrate to the paper. The paper just gives it a little bit more sturdiness. And once it's on that paper, then you can add radio-labeled dyes or radio-labeled probes to that paper. And it will light up on imaging or X-ray. So you have a paper that looks like this, and all those separated DNA segments will light up. That is your southern blot. Now you know why they call it blot, because you just basically blot it. Your northern blot will look for RNA. The reason why this is important is because if you have a DNA sequence and you want to make that into protein, review how does that turn into protein. It has to make RNA, and that RNA becomes mRNA, and that mRNA becomes protein. And the sequence between RNA to protein, you have to cut off some introns, remember? You have to cut off things that we don't express. So if you're just looking at DNA thinking, oh, that DNA is going to express X protein, it won't, because you cut off those introns and it can express a ton of things, a host of things, depending on what it cuts out. So the northern blot looks for RNA, in particular mRNA. These are the things that already have their introns cut out. These are the things that are more expressed. That takes away some of the ambiguity of it. So northern blot looks for RNA like mRNA. Lastly, Western blot looks for proteins. Proteins are proteins. In this part, you skip the first step. You don't need to have enzymes to cut it up because it's already a processed protein. It's just there. So you skip that step. You just have to gel electrophoresis it, blot it, radio label, and you're done. There's one thing I do want to talk about. Now, the Western blot is famous because it is the confirmatory test after HIV ELISA screening. We'll talk about ELISA in a second, but if someone comes in with a positive ELISA screening, you don't just diagnose them with HIV right away. You have to confirm that with the Western blot, and that's looking for proteins that's associated with HIV. That just confirms the diagnosis. One last thing I want to talk about is the Southwestern blot. Southwestern blot. What do you think that looks for? Name's up here. It'll look for proteins that bind to DNA. Proteins that bind DNA. If that sounds foreign to you, it shouldn't. These are proteins such as transcription factors. Those proteins bind DNA, make it transcribe or what have you. So transcription factors. And I had a question that said just that. Researcher was using a Southwestern blot. What were they looking for? And then they listed a bunch of proteins. And you just have to find a protein that binds DNA like transcription factors. And when we talked about blot, we talked about adding radio-labeled probes to detect what we're looking for. The radio-labeled probes here are going to be DNA because that's what it binds. These are just nucleotides. I think that's all I want to talk about on blots. Let's move on to our next one. Our next one is flow cytometry. Flow cytometry is basically looking at cells. And you're looking at the size of the cells, what proteins they're expressing. So if they have little proteins on their membranes or what have you. And you can actually see the proteins in their granules. Probably the easiest example to think of would be your T cells. So your T cells, you have your CD4s, CD8s, all different kind of CD markers, CD proteins. And so flow cytometry can really separate them. Flow cytometry is, pretend this is the back wall, and you have a laser that's aimed right at it. And if I drop things, like if I drop this marker, then the laser will hit the marker and cause some sort of shadow or light scattering behind it. And from that you can tell what size or what's the shape, the outline of it. And if I drop a bowling ball, the laser will hit it, scatter the light. And if I drop the tripod that my camera's on, it'll scatter the light. And from different angles of lasers, you can tell what size it is, what shape it is, what kind of proteins are on the surface, and that is flow cytometry. We have a wall, a laser, and then one by one we just drop cells through that laser. And by using different lasers and different angles, we can tell all about that cell. That is your flow cytometry. So that is how it works. However, you should also be able to interpret the findings. They like to use what we call a scatter plot. So let's say scatter plot. We're taking T cells again, for example. And this side is CD3 markers, this side is CD8. And they like to break it into negative and positive. So negative for CD8s, positive for CD8. Let's say you dropped 100 cells and a bunch of them made a scatter plot that looked like this. Some of them made a light scatter plot that looked like that. Some of them look like this. Can you interpret that for me? If we looked at the left lower segment, these would be all the cells, left lower, these would be all the cells that were negative for CD3 and negative for CD8 markers. How about the left upper? These would be all the cells that are positive for CD3, so they have CD3, but they are negative for CD8. They don't have CD8. So last one, the right upper, these would be positive for CD8, positive for CD3, positive for both. And that's how to interpret it. Relatively straightforward. That is your flow cytometry. Moving on to microarray. Microarray is actually looking for a lot of different things. So we talked about looking for particular DNA and particular RNA and particular proteins. Microarray instead looks at slabs of these, like thousands of these. And there's a reason for that. There's a method to that madness. Microarray. By laying them all out there, it illustrates different things. So why do we do microarrays? Well, we can look for small things like single nucleotide changes. So we have that flexibility, but we can also look at very large changes. Do you think that genetic lineage and linkage is small? No, you're going to need a lot of DNA for that. Do you think things like trinucleotide repeats, where there's just 100 of these repeats, is small? No, you're going to need something bigger. So trinucleotide repeats, genetic linkages, you can look at things like genetic mapping if you want to map an entire genome. Mapping, these all use a ton of data, a ton of this. That's microarray. One thing I want to talk about is ELISA. We hinted at ELISA when we talked about blotting. We said that the blotting test confirms ELISA. What is ELISA? ELISA looks for particular proteins like antibodies and antigens. Just an immuno review. If you have an antigen from a virus, your body will make antibodies against that. That's your immune complex. Yeah, they always go together. That's why they're called complex. So your antigen, I will just denote as this little round thing. And your antibody, I'll denote as kind of the classical Y-shaped antibody. So we take advantage of the fact that they always go together. If you're looking for a particular antigen, just have a particular antibody against it. And if that matches, then you know you have the right thing. That is ELISA. There are many different types of ELISA, but the two main ones you need to know are direct and indirect. And this depends on whether you start with using an antibody to look for the antigen, or you start by using the antigen and introducing an antibody to bind to it. We'll start with the direct one. The direct one starts with antibodies. So you fill a well full of antibodies, and then you introduce the antigen, dump it in there, and of course that'll bind because they go together like a horse and carriage. So you introduce the antigen, it binds. Now you introduce an enzyme with a fluorescent probe, essentially. And that will bind the complex. And on the bottom of it, it has that immunofluorescent probe. That probe will turn colors if a protein binds on it. And so you add that protein, and if it turns color, you know you have this whole complex. If it doesn't, you don't. Understand? Indirect is a little different, but essentially the same thing. Indirect, instead of starting with the antibody, you start with the antigen. And by starting with the antigen, start with antigen. By starting with the antigen, you're going to want to add your antibodies. Can't have one without the other. And likewise, you introduce that enzyme with the immunofluorescent bottom that turns colors when you have a protein binding to the back. So then you add the protein, and if it fluoresces, positive. If it doesn't, then you don't have the complex. That's ELISA. Again, there are many different types of ELISA. These are the main ones. If you want to know about the other ones, it is in my notes. We have just a few more things to talk about. One of them is karyotyping. Karyotyping looks at your chromosomes. Karyotyping looks at your chromosomes. Usually your chromosomes, like I said, they're condensed, they're at rest, they don't want to be accessed. But there are particular times, like in mitosis, where they need to open up or be synthesized or be separated, etc. In the cell cycle, there's a particular phase where they're nice and open and separated. Do you know what that phase is? That is your metaphase. And in the metaphase, it's gorgeous. They look like this. They're all open, they're all aligned, they're all separated. It's fantastic. But we have to act fast because, remember our cell cycle, our microtubules will pull that apart, go into anaphase, go into telophase, create those two separate cells. So we have to act fast and stop them at this phase so they're nice and aligned and perfect for us to study. We stop them by giving a microtubule inhibitor. One of the most common is going to be colchicine. And it just blocks that. Once it blocks that, we can stain them with Giemsa stain. We might have them all mapped out now. We'll just take, for example, this is our chromosome. In that chromosome, if you want to look for a particular piece of the chromosome, what do? We introduce a radiolabel probe. That's kind of the running principle for this video. If we want to look for something, we kind of radiolabel it. So we radiolabel it, and everything checks out, then it checks out. But sometimes there are changes or mutations in the chromosome. And by radiolabeling it, we can tell if there are these changes. For example, what if we want to look for a piece right here? But instead we find, we'll just say this is chromosome 20, but instead we find it on chromosome 21. That would be translocation. So translocation is outside of where it should be. Another problem, what if we, again, are looking for this, and we see this, which is great, but we also see one up here, maybe one over here, maybe one over here. That is called duplication, or more than there should be. And then one more example, if we just go back to looking for this. If we send the probe and we only see it on one allele, that is a deletion. One of them's gotten deletion. One of them's gotten deleted. So that's micro deletion. Understand? We're going to wrap this up with gene therapy. That is introducing things artificially or deleting things artificially to a cell, hopefully to benefit a patient. There are a couple of lab techniques in gene therapy that I want you to know. One of them is Cray-Lox. Cray is an enzyme. Lox is just a sequence it binds to. So we have something like this with 2 Lox sequences on here. Then Cray will bind these two and cut them out. So this part would be gone. And that can cut out a protein you want to express, or it can cut out a stop codon so you express it more, what have you. But understand that Cray is an enzyme, Lox is a sequence it binds to, and it cuts out anything in between. That's your Cray-Lox. The last one is a little more complicated. That is going to be your RNA interference. Interference. So let's say we have our cell with a bunch of identical RNA. We'll take one of the RNA pieces that we want and turn that via its complementary strand back to double-stranded. We call that double-stranded RNA. We'll introduce that back into our cell. Now it's double-stranded. And your cell looks at that double-stranded RNA and says, what the heck is that? I've never seen that in my life. Destroys that, and also destroys anything that looks like that. And that way we can shut off that mRNA sequence that we don't want. That is your RNA interference.